I'm going to talk about phosphoproteomics, and it's mass spectrometrists have been talking about phosphoproteomics about as long as people have talked about weather, and you can talk about it, but you can't do anything about it. And that's the way it was for a number of years in mass spectrometry. A lot of people talked about it, not many people did anything about it. In recent years, we've gotten better at least doing something that's fairly consistent. But it's been a subject people have wanted to talk about. The reason is it's one of the things that in mass spectrometry you can measure that's difficult to do with another type of technique. And the kinases and phosphatases are working in conjunction with one another into some response. And so it adds complexity to the proteome, and something that you can hold over the head of your colleagues who do genomic analysis. You can't really do anything with phosphorylation there. So mass spectrometry and proteomics at least allows a way that you can target this. When you think about post-translational modifications, there are a couple of things that make this a little more difficult. One, you have to identify the modified peptides, and then localizing the site on that peptide that where you have a modification. There's one serine, one threonine in the peptides, pretty easy, but it's more difficult when you have a peptide that has 4 or 5 different possible sites of modification. And so that makes it a little more complex. Some of the issues that increase difficulty come from the fact that when we're talking about phosphorylation, we're rarely talking about something that is phosphorylated 100%. And so there's some stoichiometry that's involved. And so you're looking for the presence of a modified peptide that is modified sometimes, at some point in time, and usually some percentage. It's not fully modified. There are sequence coverage gaps. We talked a little yesterday about the peptides that we see. Mike gave a good overview of the best flyers. The best flyer peptides are never phosphorylated. I wish they were, but they never are. And so there are always regions of the protein that are difficult to cover. Even if you take a serum sample and you don't deplete the abundant proteins and you digest it, you're not going to get 100% sequence coverage of albumin. The most abundant protein in the blood. You're just not going to do it because there are going to be regions that just don't produce good tryptic peptides. On average, in a proteomic experiment, you may see 30 to 40% average sequence coverage of proteins. So if you're an optimist, there's a 30% chance that you'll see your phosphopeptide of interest. If you're a realist, I'm not a pessimist. My wife keeps telling me, but I tell her I'm a realist. You're really not going to see it, right? The deck is stacked against you, and so it's difficult because you're working with something low level, and phosphorylation is it's labile, and so we're trying to make these ions fly in the gas phase. Talk about the electrospray process and what an advance that was for mass spectrometry 40 years ago, but it was the issue of forming those ions sometimes is enough energy to make the phosphorylation come off. And so it comes off sooner than the rest of the peptide fragments. And so sometimes that's difficult. The spectra here show some of the difficulties when we fragment these peptides. And so these are 3 different phosphopeptides, the top one with a phosphoserine, the next one with a threonine, and you can see there's one major peak, and it's from the loss of the phosphate. It's actually, you're losing a phosphate and it's pulling a hydroxyl group off of the side chain, off of the serine or threonine. And so when you put energy into the peptide, instead of fragmenting along the peptide backbone to get these nice Y and B ions, most of the energy goes in kicking out this phospho group. Well, it's difficult because now most of your the remaining ion in that peptide is this loss of phosphate. And so you can further fragment that peptide, but that requires an MS3 event. So it's a little difficult. You can see down at the baseline, and I'll try to use my pointer here. You know, there are Y and B ions here, but they're, you know, maybe at the 5% level. And so this is where, you know, if you're playing odds, you head to the casino and playing odds, we're looking for a peptide that has lowest stoichiometry to start with. It doesn't fly very well. It's not on the top flyer list because we're making positive ions, and now this is a peptide that we're putting a negative charge on with a phosphate. So it's, you know, more difficult. So it's going to ionize less. And now when we fragment it, the normal fragments that are produced from a peptide are at lower abundance. So again, you know, I said I'm like the merchant of death here. You know, just keep giving bad news. If you look down at the bottom, you see, oh wow, if you have a phosphotyrosine, well, here's a little good news. Peptides that have phosphotyrosine actually fragment pretty well. That's because it's not energetically favorable to get a loss of 98, and so it's a little harder for it to lose that phospho group, and so you get better fragmentation. It wouldn't be me without giving bad news, though. But the bad news is, you know, only about 1% of overall phosphorylation is tyrosine phosphorylation. So if you're wanting to get tyrosine phosphorylation, it's you know, it really involves another step of enrichment. So more difficulties. One good thing that has been added to repertoire of mass spectrometrists through the years are some scoring algorithms, A-score, and I think this was originally developed in Steve Geege's group. And there's implementations of this now in just about every software. And so this, it's a routine that goes through and it analyzes all the fragment ion data, and it helps put a localization score. So basically it gives you some confidence that the mass spectrometry data backs up to show that a specific site is modified by phosphorylation. We use MaxQuant, and it has an implementation of this that's actually pretty good. And so you can filter on this. There are times when, you know, it may give you a 50% score, and you think, oh wow, that's awful. You know, you're grading your student's test. There's an F. But if a site, you know, if a peptide has 2 serines and the data shows 50%, it's one of those two, right? You're confident that one of them is phosphorylated. Sometimes if they're right next to each other, you know, we think about biology being very specific. Sometimes the kinase doesn't care, right? It's wanting to put a charge on the molecule to have some function. It may not change the function whether serine 19 or 20 is phosphorylated. And sometimes there's a mixture of both. You know, so sometimes it's not the fact that we can't determine where it is, it's that the data says it's both. So, you know, just because you have a low, probability of localization doesn't really mean that it's a poor result. And so it's not just a quality score. And I think sometimes people often, forget that. They'll filter on a 90%, and then, you know, you're going to lose some cases where the biology is still there. This is just a blow up to see. So it gives you the sequence, and I'll highlight it here. So in this first peptide, there are 3 possible sites. And so, you know, saying that there's a 0.4% chance that this third threonine is phosphorylated, which means it's not. there's about an 8% chance that it's that serine, so that's probably not. A 91% chance that it's that serine. I feel pretty good about that, and then, multiple cases where, this example, 99% chance that it's that serine. And so, the numbers definitely do help in the confidence of the localization. Now, what do we do to get over the problems of these low abundance in stoichiometry? We enrich, and it's not nearly as good of an enrichment, I think, as everybody thinks it is. It's not like we're using an antibody-based that's very specific enrichment. You can do that for phosphotyrosine. Cell signaling, sells a pan antibody for phosphotyrosine that's actually quite good. The worst part about it is you got to mortgage your house in order to buy it. First, you have to buy a license to do it, and then buy the reagents. It's really expensive because phosphotyrosine is really low level. You've got to start with milligram quantities of material, and so it makes it difficult. But there have been advances in the enrichment of phosphopeptides, but there was an observation early on that you could use some coordination chemistry and kind of hijack some of these interactions for phospho enrichment. And so iron was the first of these, iron chelating, and then came titanium. And it's what's shown here, titanium enrichment. And so basically your titanium, the positive charge of titanium is interacting with the negative charge on the phosphate. And so you're leveraging some biochemistry in your sample here to say, okay, we can get an enrichment. But again, it's not specific, but it does work reasonably well. There are, I don't know, hundreds, thousands of papers that people go through, and they have their protocol for enrichment. And one of the real difficulties was, one lab could have a protocol and it worked great, and you try to repeat it, and in your hands, it just didn't work. Like, you could tweak the differences, and my lab finds that we need a lot more of the chelating compound. We have to use a lot less lysate, or we have to use a lot more lysate, or we have to rinse it in a different buffer. And it's kind of like everybody's chicken soup recipe. Everybody's using the same ingredients, but the ratios are a little different, and everybody likes theirs better. And you'd read a paper and you go, I know this, they can't be doing this, because I've tried this, it just doesn't work. And so any time that there are inconsistencies, and then there's conspiracies, and this one group can do it, but nobody else can do that, so they're really not saying what they're saying. Are they being honest? And this really went on for years and years. And this is a paper that came out in 2018 that really shed some light on this. And from a chemistry point of view, it makes a lot of sense. And so they came up with a method that improved their yield of phosphopeptides. And when I first read the paper, I looked at the figure, and I have it circled in red here. So here's their standard protocol, and here's their optimized protocol. And, it's a really little peak here, a really big peak there. And when I first read the paper, I thought, oh, they've got the figure legends wrong, right? I mean, surely the optimized is the big peak. But what they found was, yeah, they could get material off, but it wasn't phosphopeptides. And what they found was that they were having DNA fragments, negatively charged, will quite happily bind to, using coordination chemistry, will quite happily bind to titanium and iron. But if they treated these samples with benzonase to digest the DNA down small enough, they didn't have the issue. And so they got much, much better results. You go back and you start looking back at at papers before, and one lab would use a digestion method where through their processing, the DNA was removed from the protein. Those labs got much better results in phosphopeptide enrichment. Labs that use methods where they didn't shear the DNA, or they didn't separate it with a molecular weight filter, they had different results. So they had to use a lot more media to get to bind the same amount of phosphopeptides, because you're essentially poisoning your resin with DNA. And so this, okay, this helps. This makes a little bit more sense. One of the kits that we use, and I talk bad about Thermo more than probably most people that almost exclusively use Thermo products. But there are two kits, the high select titanium and high select iron phosphopeptide enrichment kits. And I had used these sometime back in the mid 2000s, and they were awful. Well, it just didn't work. And I went to a poster at ASMS a few years ago, showed really good data. I asked what they were using. Say, well, these new kits. Like, oh, they must have new kits. I looked. Now this is the same kit that I'd ordered, 10 years before, and it just didn't work. So I started asking questions like, oh, it's the same kit. They changed the buffers. They work now. So, okay, well, we'll see. And sure enough, it works. And so you can do phospho enrichment. You can buy resin and do it on your own. These kits work really well. It's a little expensive, but it's within reason. And one of the things that I like about it is the coordination chemistry between titanium and iron it's very similar. You're binding phosphopeptides, but sometimes interferences in the sample can affect how one works versus the other. So with the method we're using, we bind titanium first, and it doesn't have as high binding capacity as the iron. And so if we have more phosphopeptides, they actually flow through. We bind all we can, they flow through, and then we take this flow through and we capture it on the iron. And then we combine the eluted peptides from the titanium and the iron together and do one analysis. And people ask all the time, you're separating these, why don't you analyze them separately? Because some peptides, half will bind to the titanium, half will bind to the iron. So if we analyze those separately, the abundance of those peptides is, half in each sample. If we combine them together, now there's more peptide, and it's. I'll get to a figure that shows this in just a minute. So here's an example of some of the data that we see, and this was from actually a pretty small sample input, 30 micrograms of each sample in a TMT 6-plex. That's how old this data is. And we identified 13,000 phosphosites, about 80% serine, 20% threonine, about 2% tyrosine. So it passes the realistic figure. We didn't get 10% phosphotyrosine. If you get 10% phosphotyrosine, something's wrong, because 10% aren't going to be tyrosine. It's going to be between 1% and 3% usually. And of these sites identified, we quantified over 9,000 of them. And over 7,500 had site localizations greater than 90%, and over 9,000 were greater than 75%. And so it's this was data that was probably acquired in. We haven't had the Lumos in a number of years, so this was probably acquired in 2018, 2019, maybe. So several years ago. So the data we get now is actually, it's even better than this. Our workflow, we mentioned this yesterday. We start with 200 micrograms. We request 200 micrograms of lysate for phosphoanalysis. And this is twice the amount of protein that we request for a normal lysate. But the biggest difference is if we just do a normal lysate with 100 micrograms, we're gonna, it's 100 micrograms easy for us to process. We're gonna digest it all. I'm gonna take half of it and fractionate it, and I'm gonna take a small percentage of that and run it on the mass spectrometer. We're gonna end up running about 1% of what you give us on the mass spectrometer. If we do phosphoanalysis, we ask for 200 micrograms, and we're loading all of that on the mass spectrometer. And so we're asking you for twice as much, but we're using 200 times as much. And so that shows you, what the role of enrichment. We actually take 95% of that sample and dedicate it to phospho. We're doing the lysate with only 5%. And with that, we do the enrichment, and then we fractionate and let the mass spectrometer pull it out. And TMT does a really good job for phosphoanalysis. One of the advantages that it has is this TMT reagent holds on to protons really well. And so it will help phosphopeptides that are a little difficult to ionize because they have negative charge on them. It gives another spot to put a proton on the peptide and make it ionized a little bit better. It also helps the fragmentation a little bit better. And so it really is a pretty good method for quantifying phosphosites. One of the things that we can do in the analysis when we do phosphopeptides, it's a little different. The output looks similar to the output Dennis was showing earlier for the proteins, but we do it at the peptide level. And so we're looking at the individual sites. And so we can look now at specific peptides. We can take peptides and look at a motif. And so this is actually really nice because you can pull out, in an upregulated setting, you can pull out and look at certain motifs, track those back to maybe a kinase of interest, and you're able to kind of take that next step. And so there's lots of data that's generated in a phospho experiment. We have yet to have somebody, say, okay, I've made it through all the list of all the phosphopeptides, and I don't know what to do next. Usually they say, I don't know what to do next before they get too far, because if you've got 10,000 phosphorylation sites, the first thing you're going to do is look at the one site that you're really interested in. If you're really interested in that one site, probably better to contact Mike and do a targeted experiment looking for that one phosphopeptide, right? But it does allow you to generate, a lot more information. Sure. I'm sorry?
Would be similar, for example, or ELISA for phospho versus we normalize to the total,
Right.
non-phosphorylated to see that related abundance.
Right.
So is it possible with this approach, or you just do?
Yeah, so it's all normalized. Yeah, so it's normalized. It's normalized to the total protein. Yeah.
It's not phosphorylated for.
Rick, can you
Yeah.
repeat the question? We can't hear
sorry.
those.
The question was how the samples were normalized, if they were normalized to the total protein or to the non-phosphorylated form. And so we normalize to the total protein. I mean, you can look at the abundance of the non-phosphorylated form, but it's difficult to directly compare the intensity of a non-phosphorylated peptide with a phosphorylated peptide. The non-phosphorylated peptide is, it's detected in a separate experiment. It's detected in the the experiment where we're looking at the just the protein. The phosphopeptide, we've used, 20 times more material and gone through an enrichment. And so you can't compare that intensity to the intensity of the unmodified. And even if they were detected in the same experiment, you really can't use the intensity of the both to compare, because the phosphopeptide is going to ionize at maybe 10% of the rate of the non-phosphorylated, and so that's more difficult. It's a much better comparison to compare the overall the starting protein concentration, and within TMT that's easy to do. This is a paper from the Broad Institute that is the best example of phospho-specific signatures, and they went through the literature and found specific sites of phosphorylation and correlated them. And so a lot of the pathway analysis software it may have a phosphorylation, but it may not mean what you think it means. There may be one protein that has multiple phosphorylation sites. And even, if phosphorylation is up on that protein, maybe one site where the phosphorylation up increases activity, and maybe another site where if that's phosphorylated, it decreases the protein activity. And so ingenuity is probably the worst about this. You can get an ingenuity pathway with, phosphorylation. What does that mean, right? What site is associated with that? And that information is not there. And so this is a good resource to use to follow up your data. And as difficult as it is to generate the data, the real difficulty in phospho is, what do you do next? How do you use this data? And it's difficult. Like there's not a good answer. If it fits within your experiment, if you see the result that you expect, it's great. You're going to see a lot more than that, right? There's a lot more crosstalk across phosphorylation than what we realize. If you knock down a kinase and do a phospho experiment, you're going to see changes, and you're going to see some phosphorylation down, but you're going to see a compensatory reaction, and you're going to see other phosphorylation up. That network is just extremely complicated. And it's, I'm a merchant of death. So, I'm not discouraging you from doing a phospho experiment, but it is oftentimes an experiment that will generate a lot more hypotheses than it will answer. And there's a lot of wealth of information there, but it's a lot. And we have a colleague at St. Jude's who's, almost actively encouraging his researchers not to do it, because it's a lot of work. They generate, take weeks to generate the data, and will identify 30,000 phosphorylation sites. And then people sit on the data and don't know what to do with it. It's like, that's a lot of work. We could have been doing something else that you would move forward on. And so, I think that it has to be in the right case. Sure. Yeah.
So, what the photon generators?
This was a previous version of our analysis. Do we have this ability in ProteoDA now, or was this was in the
Yeah, but what was the ProteoViz?
This was ProteoViz, yeah, which is also on GitHub, but
Yeah.
there's lots of software that you can take the sequence of peptides and generate the generate.
For example, how do you read this figure?
Oh, that's just showing that of all these peptides, they shared in serine in the 4th position, and most of them had a serine, so, 20% of them had a proline after the serine. There was a high percentage that had acidic residues after the serine. The second one's a better example, is like, that site, for B, that's a much smaller, sample set, but all of those peptides had the recognition site of QSP. So, every one of those phosphorylations were, had a QSP, so that kinase is directed towards that site.
So C is an indicator that that's prefer the serum more than reading, they follow by protein.
Yeah. 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 So next year, this talk is probably going to be different. We use TMT for phosphorylation now because it's our best way to give the data to a researcher in a format that has the phosphopeptide, the site localization, and intensity of that site, to use in further downstream. The combination of phosphopeptides and DIA is growing, and the analysis software is a little lacking. And so we're able to generate, similar numbers faster for phosphorylation, but the analysis software is not quite as good. It's easy in a TMT experiment to compare, because remember, these are the ones that have the individual labels and they're mixed together. So in one scan of the mass spectrometer, you can really read out the phosphorylation differences on that peptide. And so any artifacts from the enrichment are really kind of thrown out the window, because however variable that enrichment process is, and remember, it's not a true enrichment, we're just kind of hijacking some coordination chemistry. However irreproducible that might be, in that one experiment, it's the same for all those peptides. And so their response is going to be the same. For DIA, we have to do these enrichments individually. We don't get to mix the samples together. So there's more variability in the enrichment step, combined with a little more variability in the analysis. And it's not, there are good published reports, but it's not quite ready for prime time. It's close, and people are doing it. But for us, it's probably going to take another 6 months to get comfortable with that workflow. The advantage is you're not going to be constrained to the limits of a TMT experiment. So kind of a small sample set. You may have 100 samples that you're able to do. I don't know what that is, Dennis. If you had 100 samples or a time course or a dose response that you want to look at phosphorylation, that's really difficult to do with TMT, but with DIA, it's a much better fit. We can do that.
So after you identify phosphopeptide, it has your sites that the same conventional pattern. Do you have a way to put the kinase of phosphate to in vitro validation, such as get the peptide synthesis by someone to adding phosphate? Is any company working with proteomics
to make a
validate the sites?
You can, yeah, I mean, you can have a peptide made with a phosphorylation. Yeah. 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 Thermo, so I'm talking bad about Thermo, but Thermo has a pretty good peptide synthesis group. And Mike may know, I don't know if peptide 2.0, I don't know if they make modified peptides or not. Mike, do you know?
I don't know. Because I'm just getting plain peptides. I don't even need mine purified or quantified. I just need them qualitatively to be in there. So I wouldn't be surprised that they would, but
Yeah.
my guess would be that they would, because most of that peptide synthesis is just a matter of putting in, just
Right.
a different type of amino acid at that point. So,
Yeah. But
it's no after the fact modification. It's just whether or not you can insert the modified peptide there.
yeah, I know that, I know Thermo does because they sell part of these in what they call a SureQuant kit. So you can look at the MEC Earth pathway and they have the labels, carbon-13 labeled peptides for those specific phosphopeptides. So I know Thermo does. They're not cheap. But this is just showing a paper. And again, it's a couple of years old, but it's a pretty good place to start if you're interested in looking at phosphopeptide, the literature. This is a good jumping off point for that. So I'm going to talk just a minute about a project that I was involved in with Art Solomon at Brown. And Shin was his graduate student, and just an outstanding graduate student. He came to one of these meetings, and just from his questions, and I was like, oh wow, this is really good. And I knew he was from Solomon's lab, and so I thought he was a postdoc. He was a first year graduate student, and I was wow, he's going to turn out to do good things, and he really did. He went into industry, but one of the things that they developed was a way to. They're interested in tyrosine phosphorylation, and so there have been a couple of published reports about using SH3 binding domains to enrich for phosphopeptides, because you could enrich a similar you get a similar enrichment using these SH3, called super binder domains, for enriching tyrosine phosphopeptides, as you get very similar results to the gold standard pan antibody from cell signaling. And so they did a publication where they did a comparison. The advantage is with this super binder domain, you can grow it up in your lab, and it's for pennies on the dollar. Do this pull down. And so they had the ability to enrich these peptides. We were doing some TMT analysis of phosphotyrosine, and the levels are just so low, right? I mean, you're starting with a lot of material, you're enriching, but it's still really low level. And at the time, people were using TMT and starting to do single cell or small populations of cells, and using a few of the channels to boost the signal. And so you might have 10 of your TMT channels be single cell or 100 cells, and then in your 11th channel you put in 1,000 cells or 10,000 cells, and it allowed you to make the peptide more abundant. But then when you got the labels, when you went to fragment and do the quantitation, you didn't care about the level of the channel you'd added in to kind of boost the signal. You were only interested in those individuals. And so Art and Shin had the idea that we could do this with phosphotyrosine. We could take a cell and treat it with provanadate to inhibit phosphatases. And if you do that, you kind of artificially raise the level of phosphotyrosine in that cell. And now you've got a cell lysate that has 10-fold or higher levels of phosphotyrosine. And so we did that and went through a series of experiments and showed that it indeed did work, and allowed us to identify and quantify more proteins. And it's shown here in this MCP paper. And I helped them, Sam McIntosh and I helped them kind of develop the method. And so they were nice enough to include us on the paper. And so, but it's it is a nice way of enriching for phosphotyrosine. We keep saying this may be something that we add, and at some point we likely will. to grow up the super binder in our lab and use it as a way to enrich phosphotyrosine just because of cost. Because it's so much more cost effective. But that's all I have for that. I'd be happy to answer any questions anybody has about phosphorylation. And I didn't run long, so that was, I am notoriously long-winded, and so.
Hey, Karen said you'd be 12:30.
And maybe it's because I'm standing and I want to sit down, and so maybe that's what it is.
With the, last talk, third talk, I was thinking if, the difficulties in abundance and all those details, if you're interested in a specific group, like a pathway
Mhm.
of, proteins that are affected at phosphosites, then will that be advantage? So let's say I set an example and say, I'm interested only in this pathway or this, that.
So the question is, if you're interested in a specific pathway, specific proteins, if you tell us, when you submit the samples, does that help us? It definitely in the targeting aspect, right? Because then you can target those and go after it. Otherwise, it's not going to help unless there's some way that we can use that information to process the samples. And so let's say that, you're really interested in nuclear transcription factors. That is that's what you get excited about. Okay, well, don't send us a cell lysate. You isolate nuclei and send us nuclei, because then at least we've gotten rid of the cytoplasm and cell membrane, and we're working with a nucleus. And so that's a way that you can go down. I think there was somebody interested in mitochondria the other day, I forget, but same thing. So, isolating mitochondria, and those are isolations that we're going to let you do. We're not going to do subcellular enrichments. There are kits that work, and there are lots of kits that don't work. I'm not going to mention thermo, but that's something that is that you can do in your lab, and then send us those those enrichments, we can do. But for specific proteins, one thing that can be of use is, okay, you say, I'm interested in insulin growth factor 6. Well, we can look in, mammalian cells, we can look and say, okay, we've seen this before, but when we've seen it, we've seen 8,000 proteins more abundant than this. And so by that, we know in order for us to see it, we're going to have to run this on the most sensitive instrument we have on a little bit longer gradient, because if we run it on the older instrument, we're not going to see it. The protein depth is not going to be there. So it can be useful for kind of designing what we want to see.
Two questions. Number one is, because of many of those phosphorylation, especially my research interest, we are looking at the patterns. So this pattern typically serine greening separated by 2 amino acid 3. So they're like a multiple at the same place.
Mhm.
So according to you mentioned that those cluster phosphorylation sometimes harder to get a proteomics because they cover up with each other.
Yeah, so the question is, when you have multiple phosphorylations, and we certainly see peptides that are multiply phosphorylated, but it is more difficult than a single phosphorylation. But oftentimes that's because the signal is split, and you'll have, if you've got a serine threonine residue 10 and 13, you'll see one peptide with serine 10 phosphorylated and not 13. You'll see 13 phosphorylated and not 10. And then you'll see them both phosphorylated. And so you've got, now you've got that signal spread across multiple channels. And so that's the, difficulty is that there's just more permutations to see that. Yeah, but but we definitely, we definitely see that. And that's I think one thing that's going to be nice as we get into doing phospho-DIA, you can look at earlier time points and tease out some of these activities of which happens first, right? So there may be an event that you see that, both are phosphorylated to have some effect.
Yeah.
But, you might be able to do an experiment with literally minutes or seconds at a time point, and you might see that the serine 10 always occurs first. And that, that then is recruiting another kinase to come in, and so I think breaking out, and right now it's difficult to do those because you've got, it's difficult to get lots of time points to fit in within a kind of restricted TMT experiment. But, when you have the ability to do 30 or 50 different time or dose response, I think, the data is going to be massive, but I think that's exciting.
So for that, probably the polymer, they detect the peptide individual assay.
Mhm.
Follow the trace time, and then you can do a mutation.
Yeah. So following up these with mutations, the biggest problem is in most, large scale proteomics experiments, if you identify 10,000 sites, no one's going to do 10,000 mutations, right?
Second question is, because of many phosphorylation, such as those promote the proteol degradation.
Uh-huh.
So they have in the cytoplasm, not in nuclear. So when they're not degradation, they go nuclear. So definitely make a different meaning between proteomics with the cytoplasm portion of the lysate.
Yeah.
So
So
in that case, is your proteomics able to work with trademark X100 rather than SDS lysate, for example?
so the question I think is about tracking phosphorylation between subcellular localization. So
Yes.
something gets phosphorylated and it transports either into or out of the nucleus.
Yeah.
And so the question, can we look separately
Can we
at?
look specifically interested, for example, cytoplasmic vision portion of the lysate? So you don't want to get a nuclear contamination.
Yeah, I mean, you can definitely do a cytoplasmic,
Yeah, also we are using
enrichment or reduction of other organelles and look at cytoplasm versus whole cell lysate versus nuclear. Yeah, we've done those experiments many times.
I sent you the cytoplasm, it's actually the cells trash all the nuclear, only the cytoplasm enrichment.
Mhm.
With all the protein phosphates, you have the preserved and then send you the lysate, just that part, so which will present a Triton X100, not the SDS.
Yeah, so I mean, it could work. We'd have to talk about what level of Triton, but we can handle any detergent. We just have to know
Yeah.
what it is, and
Yeah,
so.
I'll give you precisely portion what's the components in my lysis box.
Yeah. So the question, can we handle samples where you've used like Triton X to isolate a cytosolic component? Yeah, we can. With that, we do like a chloroform methanol extraction to remove the non-ionic detergent.
Rick, I've got a question based on those two.
Okay.
If you enrich for subcellular fractions, do they still need to send 200 micrograms? Like if they're doing,
That's a good question. I think send as much as you can. So the question, if you enrich for subcellular fractions, do you still need to send 200 micrograms? Send as much as you're comfortable with, because you're going to get more data the more you send. For a lysate, that's not true. If you send us a milligram instead of 100 micrograms, Lisa in the lab is probably just going to talk bad about you because you sent too much. It's not going to help. If you send that for phospho, it will probably help. The more, the better, really, for phospho. And so for that, I would say within reason, just the more, the better. That's a good question.
You got any other questions, Karen?
no, I don't think so.
Okay,